As we continue our study of the central nervous system and how it works, we will now discuss specific types of cells, what they do, and how it relates to current research. Glial cells. There are three main types of glial cells: astrocytes, oligodendrocytes, and microglia. Astrocytes are star-shaped cells in the central nervous system that have multiple branched processes. In a healthy situation, these cells occupy a large amount of space in the central nervous system to provide structural support for the neurons. They are essentially the scaffolding of the central nervous system and hold neurons in place. Oligodendrocytes are cells with multiple arm-like processes branching from each cell body that have the ability to myelinate neurons. The processes wrap themselves around axons, covering them in myelin, allowing proper communication to take place across axons. The oligodendrocyte shares a lot of similarities with their cousin the Schwann cell, the myelin-producing cells of the peripheral nervous system. They share the same function of myelinating neurons. Where it gets interesting is that Schwann cells can regenerate and additionally guide the regeneration of axons by creating a pathway for them to regenerate within the comfort of myelin. This makes Schwann cells a key target of research to regenerate damaged axons after a spinal cord injury. Microglia are macrophages, tiny star-like cells within the central nervous system. They are like the garbage men of the brain and spinal cord. Their job is to clean up cellular debris. They scavenge for damaged cells and other harmful invaders to engulf them and kill them. What happens after a spinal cord injury? A spinal cord injury can be divided into three phases: the acute phase, seconds to minutes following injury; the secondary phase, minutes to weeks; and the chronic phase, months to years after injury. The biology of these phases play an important role in determining functional outcome. Initially, after the spinal cord has been injured, there is immediate cell damage where some nerve and supporting cells like glia are either injured or die. The mechanism of the injury causes this physical damage. This marks the acute phase, and immediately neurons stop signaling below the site of injury, a process known as spinal shock. Next marks the secondary phase, the most impactful stage on functional recovery. The initial trauma to the spinal cord causes a chain reaction of inflammation and release of toxic chemicals by the damaged cells, which kill or demyelinate healthy neurons and supporting cells in the area. In the secondary phase, microglia become activated and form into macrophages. Macrophages are the cell types that cause inflammation, which is a normal response to infection or injuries. Also during the secondary phase comes the formation of the glial scar. In the event of injury, astrocytes multiply and move to surround the area of injury in order to protect the surrounding tissue. This wall of astrocytes that is built to separate the healthy cells from the injured cells is called the glial scar. The glial scar helps protect the surrounding tissue from damage, but it also inhibits nerve cell regeneration. The astrocytes and macrophages that make up the glial scar produce a molecule called chondroitin sulfate proteoglycans, CSPGs, which surrounds the site of injury and inhibits neuronal growth. As the damaged tissue and cells within the glial scar gets broken down, a fluid-filled cavity or cyst forms containing inhibitory molecules that create an environment that prevents nerve regeneration. In essence, the cavity creates a physical gap and hostile environment that blocks axon regrowth, whereas the glial scar acts as a barrier and contains substances that prevent nerve regeneration. As we'll see later, one area of research involves dissolving the glial scar to allow for neuronal growth to begin again at the site of injury. Within months of spinal cord injury, the secondary phase of injury subsides and the chronic phase begins. By then, the inflammatory response and neuronal injury has, for the most part, stopped. The chronic phase primarily results in some natural regeneration of nerve cells, which often leads to secondary complications of pain and spasticity. Currently, spinal injury research is focused on 5 key principles for spinal cord repair: neuroprotection, protecting surviving nerve cells from further damage; restoration, dissolving the glial scar; cell replacement, replacing lost and damaged cells; regeneration, stimulating regrowth of axons; retraining, using intact circuits to restore function. Advancements in any of these key areas would mark a significant step towards a cure for paralysis. In our next video, we will look at how scientists are developing ways to protect the injured spinal cord from further damage.